Okay. Thanks.
Oh,
Thanks for the intro.
sorry, that was really short intro. So that's Jonathon Vandezande from the Rowan Scientific, and he will have a tutorial on how to use Rowan.
Okay. Um, yeah, just trying to. Okay, can everybody see my screen? I'll take that as a yes. Um, Hi everyone. I'm Jonathan from Rowan. I'm the director of computational chemistry here. We are building a computational chemistry platform that's kind of modern and on the web. Corin gave a talk yesterday about what we're hoping to build at Rowan, and today I'm going to be giving a tutorial on how to use Rowan. So I guess before we start, you can go ahead and make a free account if you'd like to follow along. You can just go to RowanSci.com. And if you can just mention 9ad in the Heard About Us section, just so we can better keep track of stuff. Um, so for this tutorial, we're gonna go over seven different things. So we're gonna just start with how do you create projects and manage your data. I think this is a really important thing to consider with computational chemistry, just given how much data people have. Um, we'll do just a quick basic optimization. This is kind of what you're used to with most computational chemistry programs, and just kind of using this to get you started. Then we'll walk through things like conformer searches, scans, reactivity modeling, pKa, and finally do some virtual screening. If there are any questions along the way, please feel free to raise your hand, and I'll try to get them as possible or put a message in the chat. Um, so let's get started. So first thing you should do is log into RowanSci.com and start making a new project. So you can click the Create Project button. We'll type 9, add here, and create it. Um, you can then go to the structures part, and you'll see a bunch of different things, including pipelines, which will be released soon, as well as structures. And here we can add a variety of different structures that we're interested in our project. Um, and so we can draw in 2D, we can draw in 3D, we can paste our XYZs, we can even do fun stuff like inputting from smiles or inputting from a name like PubChem. So for our stuff, we'll be dealing with Elixir. That's probably spelled wrong. Nope, I got it right. Okay. You can see here's the 2D structure. I can convert it to 3D. We can go ahead and save this to our thing. We can now switch to the folder view and begin our calculations. Here is kind of the main view of running calculations in Rowan. You'll see there's many different buttons like here to do a variety of different modeling tasks. We'll just start with a basic calculation. Here you're again shown this view where you can either upload existing files that you have, maybe from some other program. You can draw 2D or 3D. You can again input from a variety of different types as well, such as smiles, PubChem, or from our project structures. So since we already imported Elixir, we can go ahead and click on that here, import this, and we can see our molecule. We can rotate it in 3D. We can edit it if we want. We can say add a isopropyl group here, and we can add ones, and we can now just like look around again, save it, or we don't want to save our edits. We can then choose whatever level of theory we'd like to use for our optimization. So the OMO models are pretty good. AIMNet2 is nice and fast. GFN2XTB is a great semi-empirical model. And then we also have a bunch of DFT methods as well. You can go to more methods to select things. We'll just start with AIMNet2, and we'll run an optimization. Uh, this will be all uh, running on CPU and should run relatively quickly. We spin all these nodes up in the cloud, and so you shouldn't have any significant waiting time. Sometimes it'll say queued because we're just spinning up a node, and so that'll usually be 30 seconds to a minute or whatever just to get the node running. But there's not actual like traditional queuing time like high performance computing servers where they're oversubscribed and you have to wait for new nodes. They're functionally unlimited on AWS. We do cap how many you can run at the same time just to help protect you from, say, submitting accidentally 1,000 in a row and burning up all your credits. But yeah, okay, we can see our optimization's done. Took 120 steps, and we can kind of visualize how it changed throughout the optimization. You can see it shifted a little bit, but it was already in a fairly good geometry. Traditionally, also, I would recommend running frequencies to make sure you're actually at a minimum. Frequencies take a little bit longer, but they can be pretty cool to look at because you can visualize various different IR modes and such. Um, so yeah, that's basic optimization. We can now look at a bunch of other different things we have. We can do stuff like conformer searches. So here we'll just draw a nice simple molecule in 2D. So you gotta load our 2D structure editor, and let's just do pentane, nice and simple. Here it's in 2D. We can convert it to 3D here. We can see our molecule. We are using ETKDG in this case from RDKit to do stochastic generation of potential conformers, and then we use AimNet2 to clean up these conformers because ETKDG usually produces a wide variety of conformers, but a lot of them are high energy, so we need to screen them out. So we can go ahead and submit our conformers. This here it says queuing because we're having to spin up a node for it, but it should run relatively quickly. The nice thing is, while these things are queuing, you can always go on to uh, submitting another job. Here we could say potentially resubmit this pentane as a scan if we want to like look at the effects of how a dihedral angle will uh, changing a dihedral angle will affect the energy. And so let's do a whole bunch of steps. And this will run on a T4 GPU, should run fairly quickly. That's a nice thing about running on these GPU instances. And so we can see here, um, it's already running. And so we're going to scan around this dihedral angle. And so this will update usually about every 10 seconds or so is the polling. And so then we'll see steps slowly come on. You can see here as our first two steps of the scan, fairly small energy difference. So it may look large just because these are the only two steps, but this will quickly start looking more like what you're used to. We can click on a specific step to see how things are. And then as things run, they'll continue to add more and more steps. We can also animate this movement. You can see here the dihedral angle changing. We can also see that our conformers has run here. So we can click over to conformers. It looks like it's run all the various different conformers. And as we expected for pentane, the linear pentane is the lowest in energy. And then the next lowest energy is the gauche here.
It's not. They have locked it. I see.
Sorry, I can't hear the question. Is there a question? Okay, I'll take that as a no. I'll keep going. Yeah, we can kind of flip through all the various different conformers. Here we can see that we've rotated the back methyl group. And such. We can go back to our scan. Let's go ahead and rename this to something more useful. We can rename these as well. So now we can see much more complete scan here. of the dihedral angle. And we're done. Yep. Yeah, if there are any questions, feel free to post them in the chat, as Carol mentioned. Yeah, so that's how you would perform a conformer search or scan. You can obviously do this with significantly more complicated molecules. We also have a bunch of different models for either faster or more accurate, depending on what you're looking for. But that's kind of the intro. And now into my favorite part, which is reactivity modeling. Oftentimes, if you're trying to find a reaction, you're looking for a transition state. And the traditional way of dealing with this is to kind of hand draw your reaction and then run a transition state optimization, inevitably fail and continue going back and forth. Sometimes you'll say do a scan over coordinates, but one can imagine with, say, a Diels-Alder reaction, trying to run a scan over the Diels-Alder coordinate can be incredibly difficult. A lot of what I did in my grad school was just finding ways to find transition states. So one of the things I'm really happy about what we've developed here at Rowan is our double-ended transition state search method. These still don't always work, but they usually work reasonably well. So we can draw our molecules in 2D or 3D. Let's go ahead and draw today in 2D. We're going to show just a relatively simple reaction, but one of my favorites, which is just water plus isocyanate. This is kind of a base reaction that's very common in polymer chemistry. So we have a water here and an isocyanate, which is just this. We can save this and convert it to 3D. And we have, here's our 3D structure of our starting materials. We can just run a quick optimization to get them into somewhere reasonable. You could also, if we wanted to, we could shift things around. We can move things wherever we think is reasonable, perhaps rotating things around. But generally, I find in the double-ended transition state search, we don't need to have a perfectly good starting geometry. Even a geometry like this usually is good enough. We'll set the AimNet2 model here. And then what you need to do is once you've drawn your reactants, you also need to draw your products. Usually my favorite way of doing this is just copying the XYZ structure and pasting it into the product section and then editing from there. This makes sure that you have consistent atom ordering. So we'll look at it here. And then we need to just change these bonds. So what's going to happen is in this reaction is we're going to break this bond. We're going to form a new bond between this hydrogen and nitrogen. And we're going to form a new bond between this oxygen and carbon. And what we can do is we can just use the optimize with force field to kind of quickly snap it into place. In this case, I know that we usually have this all the way up here. And so this is going to be our optimized products. We can quickly do a GFNFF optimization if we need to. There's a lot of minor tricks to get things to go well, but here now we have both of our reactants here up top and our products on the bottom. We can then call this and submit. So now what's will happen is it'll run the freezing string method to kind of draw the reactants towards the products and the products towards the reactants. So they'll slowly go up the hill of the reaction. And hopefully we'll quickly see this. At the moment it's queued. There's of course many other ways one can do, and one can deal with much larger systems as well. They'll run a little bit slower just because your gradients take longer. But actually, I find for larger systems, FSM tends to be much more stable because you have just have how the interpolation works. It tends to work better for large systems, which is kind of funny. Yeah. Any questions about reactivity modeling? Nope, everybody understands this all perfectly. Uh, okay, so it's running now. It's getting our initial steps, doing the interpolation, and in a little bit we'll see the actual stuff. Oh, yes. Good question, Carol. So this works perfectly fine for transition metals. There's no actual bonds that are coming into play here. This is purely doing an interpolation in internal coordinates, and so it can work perfectly fine for stuff like that. Um, Obviously, AimNet 2 won't work for this. You'll need to use a different model. So you can use DFT, that's going to be incredibly slow, or you can use the OMO models, and I found the OMO models work really, really well. And so we can see we've already got a couple steps here. Obviously, it jumps pretty quickly across. We can see it's kind of already following along this coordinate. And so you can see we've projected to something that looks kind of like the transition state. And then what will happen next is it will pick the highest point and then run a transition state search from that. And it looks like it's already done that. Let's hope it worked. Looks like it did. Okay, so here's our optimized transition state for this. We can look at the underlying calculation. So it started here from this point and then ran a transition state optimization, slightly adjusting everything. We have the frequencies here, so we can confirm that, yep, this is the actual reaction we were looking for, where we have this proton transfer at the same time as we're getting the oxygen attacking the carbon. And we only have a single imaginary frequency, which is good. So that means that we have hopefully what we were looking for. We can go back to our previous calculation. We can view the optimized, and we can then resubmit this as a IRC. So this is typically how you will confirm that your reaction actually does connect the reactants and products, and we can go ahead and submit this IRC. And so this just starts from the transition state and then either goes forwards or backwards along the reaction path. I've got a longer blog that I can share that goes into more detail about what's happening. Um, Yep. So the transition state is running in the back end. This is using geometric to run the IRC. And then, of course, we're using AimNet 2 again as our model, just because AimNet 2 works nice and quick. And so you can see we've already got some steps. And yep, that looks like the transition towards the product. And then we can look here. We're looking like we're going more towards the reactants. And it'll just slowly add more and more points. We can go ahead and see the whole path. Now, it should be important to note that the ends of the IRC here are not necessarily the starting and ending materials, right? We don't necessarily have the truly fully optimized reactants and products here. It's just because we limited it to 30 number of steps. So you can't necessarily look at the energy barriers on this IRC to give you your results. I've seen that mistake a couple of times in papers. But it will give you kind of a general idea of what's happening. Also, sometimes since forwards and reverse is arbitrary for reactions, you may need to flip the x-axis. In this case, it aligned with our vision of what reactants and products are for this reaction. But other times you'll need to flip it to make things look right. You can also add some notes to your thing, so we can just say and so we can save this, and this will be exist then for future. Any more questions on reactivity modeling? Okay, we can go on to the next thing. Feel free to continue to ask questions if there's anything that comes up. So next we have PKA. So we have two different ways of predicting PKA. We have microscopic and macroscopic. Microscopic PKA was kind of our initial PKA implementation, and this is where if you have a single product center and you add or remove it, you can use an AIMNet2 model and a nice linear regression to get a quick estimate for the PKA of that center. However, if you're dealing with multiple different product centers, you can run into problems with using a traditional microscopic PKA prediction. So today I'm going to show us macroscopic PKA and some of the fun things that happen there. So we're going to use succinic acid. So succinic acid is actually a very interesting system because it has two similar carboxylic acids. And we can run this calculation. We can also do things like computing salvation energy and blood-brain barrier permeability or aqueous solubility based on pH. And so we can go ahead and submit those. This usually runs relatively quickly, usually 30 seconds to a minute, reasonably independent of system size, but it is highly dependent on the number of product centers you have because you can develop a whole bunch of different microstates if you have lots of product centers. Um, yeah, so as that is running, are there any questions about PKA?
No
Okay.
questions? Oh,
Did I hear something?
do you hear us well?
I'm having a hard time hearing with that mic in the lecture room.
I see that there are nobody, but the microphone is should be here. You should hear better. Yeah?
Yeah, okay, now I can hear you.
So yeah, if you have questions, you should go forward just to ask it. But currently there is no question, so you can move
Okay,
forward.
great. Yeah. I'll drop in the chat a link to macro PK versus micro PK. This is written by Corin, our CEO. He did a great job of kind of explaining why you need this. But here we can see the interesting state for succinic acid. So we have our usual fully protonated state 2 here at really high pHs, and then we'll see that tends to drop off. And then we have a monoprotonated state 3, and then a fully deprotonated state 1 here, and we get two different pKa's at 3.8 and 5.09. And this is really interesting because we have basically interactions between these different product sites. If you can do a fun little experiment where you take increasingly larger dicarboxylic acids and you can find that these two transitions slowly approach each other. Yeah, and so we can do this obviously for much larger molecules. This one, let's see how long did this take to run. This took 30 seconds. It all runs on CPU. Obviously, you can get a combinatoric explosion if you start putting in like 10 different product sites, because then it has to consider all possible one, two, three, etc. Deprotonated systems. So, yep. So that's PKA. And so this is really useful, say, for preparing your ligand for eventual virtual screening. We also have a variety of other different workflows that may be of interest that I can always go into later. But for now, I think we're going to just go ahead and jump into the virtual screening section. So for virtual screening, we have a variety of different methods, kind of protein ligand stuff. We have your traditional docking, the much newer co-folding that can also give you binding affinity. We also have various different MD related stuff, as well as batch docking, which allows you to dock a large number at the same time. We'll be coming out with analog docking soon, which allows you to dock similar molecules together so that they have kind of similar conformers. This is really useful ultimately for feeding into FEP. So we have that coming out relatively soon. At the moment, we're in beta with a couple customers, but hopefully we'll see about that maybe in a month or two. It'll become more public. So we won't go into detail on that, but just know it's coming. So we'll go ahead and start with docking. So for docking, actually, let's go back to our structures. And we want to save a new structure. This will be saving it to our system, and we want to add it by PDB ID. So 7XRR is the one we're going to load. This is a protein implicated in narcolepsy, and this is why I uploaded alorextin earlier, because we're going to try to dock alorextin into this. We can go ahead and sanitize the protein. This helps get rid of unimportant stuff. So we want to save our current pocket here that looks like there's a ligand in there. We'll save that for redock. And this will just make our future easier. We can see here we now have a pocket defined based on an existing ligand. And then we need to sanitize this. Sanitize can remove kind of unresolved residues, random solvent molecules, random other atoms, etc. Usually takes a few seconds to run. And you'll have to do this every time you want to run a docking. Looks like it's already done. Great. Now we have here our protein structure, and we'll go ahead and create it. So now this is saved to our project. We can see we have a molecule here, and we have a protein here. We can view it to show that, yeah, that is the target one. Then we'll go back to our root folder and start running calculations. So let's first just start with a docking calculation. We'll upload this from our project structures here. And we'll upload alorextin. We can redock then from a saved pose. We can blind dock, which just like throws it randomly at the protein, but it takes a lot longer. We can also manually define our docking center. We can kind of change how accurate and fast we want to do it based on various different docking settings. We can run a conformer search ahead of time, especially for complicated molecules. This can be really important to feed into docking. And then we can also do pose refinement as well to give us kind of a strain prediction. So this one is predicted to take about 3 minutes. It's a fairly demanding job. It needs to run on CPU, sadly. And so we'll just go ahead and get that one started.
How do you prepare protein structure in your pipeline? Do you have some automatic fixes, curation?
Yes, yeah. The question is, how do we prepare our proteins? So there's a sanitization step that I did, automates a lot of what needs to be done. So usually you can just upload straight from the PDB as we did, and then we'll do things like remove random solvent that gets left in the PDB structures. This is not uncommon. Sometimes there are residues that are not fully resolved, and so we'll either resolve them or we'll throw them out so that they're not an issue. Sometimes you have random salts that are left in, random ligands that are left in. Obviously, you don't want to have additional ligands in there that when you're trying to dock something new. So we have to remove those. And all this is in our pipeline. I think at the moment it's technically not exposed by our API, but we're changing that. So you'll be able to run this via API as well.
And if there are some missing residues completely, you didn't remodel them?
We do not add missing residues. That's on our list to do, but it's hard to do it in a kind of a foolproof manner. So if you have a structure with missing residues, you'll at the moment have to go in and edit it yourself.
Okay. Maybe some other questions?
So just as an example, it's an automated process, but to what degree, if people really want to, can customize this? Like even just a simple example being your sanitization procedure, or is it kind of on rails for generality?
It's both. Obviously, like we have our on Rails system that we handle all of that. But if you'd like to sanitize it yourself using any other thing, you can do that, and you can just upload your own. So if we go back to docking, you can upload a PDB file that's sanitized however you'd like. But yeah, at the moment, our pipeline does not have significant user selectable sanitization stuff. Looks like there's a question about cofactors and metal ions in the pocket. Metal ions are inherently a problem just because they're not necessarily parameterized. We're working on methods for better handling them. But yeah, at the moment, there's a lot of these force fields are not parameterized yet for metals, and so we need to do more work there. Um, for cofactors, that's again not available in docking, but we'll get into this with cofolding. You can deal with that. And I should also mention too, sometimes if you have a protein that's has say four different copies of the protein, there's an option that'll allow you to just select out a single version of the protein, and so that'll make your docking significantly faster and easier. Um, Well, it looks like the election's queuing for a while. I'm not sure why it's going so slow,
And
but
how
we'll
do
go
you,
ahead and
sorry, and how do you sample those microcycles and small ring systems during the docking?
um yeah, so this is usually where you I would recommend running so go over here, running a conformer search. And so that'll do a much better job. So there is some sampling of different ring stuff in the default docking conformer stuff. It spins a bunch of bonds, but it doesn't do a very good job, especially for things that are highly coupled. And so this is where running a proper conformer search ahead of time will really help out.
Okay, thank you.
Yeah. I'll go ahead and start a co-folding job. So co-folding is kind of the fancy new thing on the block. Um, so we'll go ahead and add this in. We'll add our ligand from project structures. And so what co-folding does is it just takes an input sequence of amino acids indicating the protein and the smiles from your structure and then generates a co-folded 3D structure. This uses some really cool stuff. You've probably heard of AlphaFold, etc. And effectively, this is doing the same thing. So go ahead and submit our co-folding. You can do a lot of different things with co-folding. There's a lot of different bells and whistles that are available in co-folding, whether you're trying to say co-fold a large number of things together, you're trying to do cofactors, you're trying to run optimizations, etc. And yeah, so at the moment our co-folding is running, and we'll go back to our docking. Looks like we have 3 docked poses in here. You can see them sitting in this little cube. We have a couple different ways. So let's just focus on pocket so we don't move around. You can see we have 3 different potentially docked poses. We can kind of edit what we see in the viewer, manage protein view. We can make it more cartoony or less. We can hide the nearby residues. And so now we can just see just purely our ligand in space, and we can look at the different poses. So we can see that we have docking scores. Usually a lower docking score is better, a more negative docking score is better. However, docking scores are not really a good indicator of affinity. It's more the case that a high docking score is bad. and indicates that a molecule won't fit. Less than does a low docking score indicate that it is good. So, we get a little tooltip there. And you can also look at pose busters. Pose buster just kind of validates the chemical and geometric consistency, making sure that it's kind of a sane structure that's generated. You don't have extraordinarily torqued different bond angles or stuff. And this is important because a lot of times the force fields that are available in docking are usually designed to be really, really quick, and so therefore they're very inaccurate. And so they kind of work in terms of telling you, is this possible to fit, but it doesn't necessarily generate very good structures. On the other hand, we have co-folding here, which has run. We're immediately greeted with an incorrect stereochemistry thing. So here in our co-folding, we always check to make sure that your stereochemistry stays the same. And one of the frustrating things with co-folding is it can sometimes flip the stereochemistry of a center, and then you'll need to add some sort of physics-based constraints to make sure that doesn't happen. But for now, we'll just ignore that, and we'll go ahead and look at our co-folding scores. And so here's our pose in here. We'll go ahead and manage the protein view to review by residues, and we can see here is our system fit in there. Gives us our predicted affinity, as well as its confidence in those. We can see it's fairly confident in this score and such. You can also look at the sequences here. Here's our amino acid sequence and our ligand smiles. This, I'd say, co-folding works generally well for systems that are reasonably well behaved and have good training data in the area. The jury is still out on how good everything is. There's lots of debates in the literature. I won't delve too deeply into them, but happy to talk after if anybody has any questions. Okay. Any questions on docking, co-folding?
Seems no.
Okay. Well, now that we've kind of developed a reasonable molecule for docking into this, we can then go ahead and kind of analyze the molecule that we have. So we'll then go ahead and look at bond association energy prediction. So if we upload elixirextin again, you can see here we have a fairly nice molecule, and we want to see, okay, where's the potential sites for degradation, and should we potentially modify it so that it doesn't have any problems with degradation. And this is what we tend to use here. We can use neural network potential, semi-empirical methods, or DFT. I wrote a paper on this a while back, and we found that neural network potentials work really, really well for this. So we'll just look at all the carbon-hydrogen bonds and break them, and then see kind of the energy that it would cost to break them. So this will be useful for predicting potentially where we would need to make modifications to our drug to make sure it lasts longer in the body. And I dropped in the chat my preprint on this. So this goes into detail on exactly how we do everything, all of the various different parameters, etc. It's a relatively straightforward protocol that you could write yourself. It's not too hard. So that's still running. There's also a lot of other things we could look at it. Our molecule, we could run, say, a membrane permeability prediction. So this will predict how quickly our molecule will permeate the membrane. We have both a neural network-based task learning thing, or we have a physics-based method. So let's just go ahead and start the multitask learning. And then we'll also resubmit this for using PyPerm. Needs to be a 3D molecule, and we'll go ahead and run that.
Are these approaches published?
Yes, they are in the process of being published. Some of these are so the physics-based method is based on PyPerm, so this is already out in the public. And then we're publishing more on the ML soon. Yeah. And so here's the physics-based one. You can see kind of the energy profile. So it's going into the membrane, it likes that, and then goes out. Right. So you have some hydrophobic and hydrophilic parts. And so we can look at various different predicted permeability coefficients, and this looks like a very nice permeable molecule, which is good.
Which data you used for training of these models, some public or also proprietary data was also used?
No, this is all public.
Mhm.
Do we need to upgrade our account to run this? It doesn't allow me to go through unless I upgrade.
Yes, correct. This is a subscriber only stuff. We figure this is pretty niche. Most people don't need this. But I guess this is probably a conference where there is a lot more med chemists who would be interested in this. Yeah, if you have just like a single molecule you need to run, feel free to message me and I can hook you up. So we can see our bond association energy prediction is running. We've already got a couple of them run. We can see kind of aligns with what we'd expect. The carbon hydrogen bond here being next to a nitrogen center is much lower, whereas carbon hydrogen bond that's not next to a nitrogen center is significantly higher. And it just will continue to populate these as we run. Yep. We also have a variety of other workflows. We have like an ML-based ADME-tox prediction, so we could throw in our molecule here. Obviously, there's already ADME-tox data out there on this, so it's going to be more accurate than would be expected from just some arbitrary molecule, but it'll give us an idea of how this should run. We also do stuff for those who say are working in materials, where we'll like do a spin states prediction workflow. You can also predict your hydrogen bond acceptor and donor strength. Here we have a couple different hydrogen bond acceptor locations, as well as one hydrogen bond donor. So we can go ahead and run this. I'll turn off conformer search just to make this run a little bit faster. And this will give us an idea then of where we could potentially modify our interactions with our protein to make it either stronger or weaker as desired.
Jonathon, there are two questions in the chat.
Oh.
The first one, does the permeability predict a flux?
It can. Those are related. This is, I mean, so efflux is a relatively complicated thing, but we can look over here. We have these are the properties that it directly predicts. So it predicts is the CaCO2 intrinsic permeability, BLM intrinsic permeability, Pampa, plasma, impermeability, and then the blood-brain barrier. So these are the things that it directly predicts, but you can make correlations with efflux.
And the second question is, do you get admit from existing values or an ML pipeline?
Great question. Yeah, so this is AdmeTalks is just running based on ML. So yeah, you can read more from these great articles. You can see citing these results. This comes from AdMet AI, which is a machine learning model for large scale AdMet prediction. And so yeah, it's an AdMet. It's not based on I mean, I guess it is based on existing values that people have measured it at some point, but it's very much an ML model.
Another question is, considering the docking, the platform generate the grid box by themselves, or it must be made mainly manually, probably.
Yeah. Yeah, so you can, I'm not sure why it's not loading. I think my internet's bad today. But yeah, you can manually define your grid box, or you can blindly dock, which will just try to basically put the box around the entire protein and just randomly throw it at that. That's really slow. You can also redock from a safe pose. So if we say redocked from our existing stuff, we could then do kind of like a finer thing. And so what's often common, if you really don't know where the pocket is, you'll just run a blind dock across the whole thing, and then you'll take the best hit from that, and then you'll redock from that posed using more accurate methods. But yeah, if you wanted to manually define it, you can also like specifically state the parameters here. But usually that's just too much work. I find it easiest just to redock from a safe pose or just blind dock and then redock.
If you have published paper on molecular docking using Rowan place from, could you share it?
We have individually not published papers on docking, but others have used our docking. I can share you. Let's see, what do we have some. Yeah, we don't have great stuff, but under the hood you can see that we use Vina Vinardo or Vina Quick Vina. And then we can use various different executables. So Autodock Vina, Quick Vina 2, QVina W. QVina W is really great for blind docking. And then two different scoring functions. And then looks like there's a question also, can you predict new druggable sites using this platform? Yeah. Oh, yeah. So this is where you'd use blind docking to find your pockets. And so this would just dock against the whole thing and then see where's a potential pocket. Obviously, there's always caveats with how accurate actually is docking. So you'd need to kind of run it up the chain of more and more accurate, such as running your docking to get your kind of guess for your pocket, and then run some sort of pose analysis MD to make sure it doesn't just like immediately fall out. And then ultimately you'd want to finish up with FEP.
And how do you perform blind docking? So you define the grid, the whole protein in a single grid, or you create multiple small grids around?
Yeah, you just put the box around the whole protein. So if we
But then the question is how much exhaustiveness you select, because to sample enough this large space.
Yes. Yeah, this is what QVina W is for. You are correct. It doesn't do a great job. This is why you would need to redock from blind docking. Yeah, right. So here, blind docking, you don't need to put your grid box down. It just goes against the whole thing. And so you can see here we have visually just the boxes around the entire protein.
Okay, thank you.
Yep. Looks like our bond association has finished. So we see we have a lot of different ones, and we can find lowest energy one, which, as expected, right, is next to this phenolic oxygen. And so that's going to make it pretty easy for the generated radical center to delocalize into this phenolic oxygen, and there's a whole ring. We can also look at, right, we got this one right next to the benzene ring as well. Here, this one is next to an amide, etc. And so we could, if we identified some of these as particularly problematic, we could perhaps swap out this carbon hydrogen with a methyl group or so. Would not want to have to be the actual organic chemist who does all that work, but if this would still then give you a good, say, docking or ultimately FEP score, then you'd be like, okay, now we can have potentially longer instance in the human body. It'll degrade slower, so it'll last longer, which is good.
Is it possible to somehow interpret these absolute values, or just as you use them as a rank?
They're just relative. I mean, so they do mean something from an absolute perspective, but generally, there's no like well-defined terms as to what is a good or what is a bad. Obviously, as someone who's looked at these a lot, once you're in the 80s, you start kind of wondering and thinking, okay, maybe we should go for something newer or for some modification to make it higher. If you're below 80, then it potentially becomes problematic, but there's no like hard and fast rules, and it's yeah, degradation is a very complicated thing. So this is only one minor part of the prediction. You can now go back to our hydrogen bond prediction, and as one would expect, we've got some nice acceptors here for these oxygens, right? We also have acceptor on this oxygen, an acceptor on this oxygen. Obviously, this oxygen is not going to be oxygen, yeah, is not going to be nearly as accepting as the other oxygens just because it is participating in this hydrogen bonding already. You can also look at our one hydrogen bond donor, but it's not going to be a very good donor because it's already interacting with this oxygen here. And so if we were trying to potentially make this oxygen better interact with a product center on the protein, we might want to say swap a methyl group in here so that there's no internal interaction here, and this one will then become much better at interacting with the protein. But of course, that'll cause some steric clash, and so it could shift the geometry significantly. It becomes complicated, so you need to end up running a lot of these. Yep. Yeah, I mean that's the majority of what I was hoping to cover today. I'm happy to take any requests or questions. I don't know how much more time we have, Carol, or do we have any idea of when this ends?
Other questions? No? Yeah, very fun.
So can I ask you about the protein binder design? So let's say I want to design a hemoglobin binding protein. Do I submit the hemoglobin structure in like the two chains or as a protein sequence? I'm kind of stuck.
Yeah, so there's our protein binder design is based on the Boltz gen. So you can read more about that if you want, but I'll quickly go over it here. So we can design proteins to bind proteins. We can bind peptides to bind proteins, or small molecules, or design single domain antibodies. And so your input's all going to depend on exactly what protocol you're using. So you can start with a sequence or PDB ID. And then you can put in your ligand, you can put in other protein structures, etc. It gets relatively complicated. And so now we have multiple alphabetical chains. Yeah. Yeah, I would say like talking about protein binder design is probably a whole class in itself. There's a lot that's going on there. I guess feel free to email me if you have questions. But I don't think I'll be able to go like into detail to a satisfying amount in this. There's just so many different things you can do. Yeah. If anybody has any questions, feel free to email me at the email I dropped in the chat. It's just my first name at RowanSci.com. Also, guess I should mention if you ever have a bug or an issue, you can always click on the little bug in the top right corner and then submit information about this. These bug reports are really useful for us. So thank you very much for submitting them. They can be really helpful for us making the platform better. So just put like a quick information about the workflow. Let's go over to a workflow so you can see. Looks funny. This will make the workflow public so we can see what you did. Sometimes this is going to be necessary so we can understand the specifics of what's going on. I guess it should be reiterated, like we can't look at any of your workflows unless you explicitly share them with us, which is great from a security perspective. It's hard for us from a debugging perspective if we can't see what was run. So if you can share the workflow with us, and this is not having any sort of proprietary information, there's no IP concerns, then we do ask that you share it with us just to make things easier. Then make a nice quick little description about, say, what browser you're using, if it's say a visual thing, if things failed because there's a metal containing thing and the workflow doesn't properly deal with metals, then please put that in there. And you can upload additional files if you want, and then submit. I won't submit and spam our things, but yeah, if you ever have any issues, feel free to submit them that way.
That's all, I think. Yeah. If there is no further questions, no further information. Yeah, if so, I would like to thank you. So, Jonathon.
Yeah. Thanks for having me.
Yeah, thank you.
Okay, so good afternoon. My name is Marian, and I really heard you the last session. I realized it's a long day, but there is much more to show you. But I hope it will be at least a little bit interesting. So what I want to talk about together with Anna is how to use our tool called P2Rank for molecular docking and for identification of panels. And so I will start a little bit by showing you what our method does and how it works. So just what we are going to predict now is a binding site. Oh, it's not good. So it's frozen. Your screen sharing is paused. Okay, let's do it again. Okay, It's paused again. How do I get from this? Any idea?
We're done.
Okay. Maybe we will have it like this, then it works. Okay, so what I'm going to talk about is a binding site in general. So what we predict is a binding site. That necessarily doesn't mean it's an active site or it's a binding site for something biologically relevant. At this moment, it's just a place which we think can bind a small molecule, right? And the small molecules can have a different size. So when I will be talking about P2 rank, our method, I don't mean very small things like ions. I will talk about it, but it's just the small molecules can have different sizes. So that's what we are trying to do. And I would like to present you the tool we have developed in the group with David Hoxha at Charles University. So what it does is it predicts surface regions, that's also important, capable of binding unspecified small molecules. So when we do the prediction, we are not saying what is going to be bound there. We are just classifying the surface residues as either binding or non-binding without being more specific. And to do that, we need a protein structure. Nowadays, there's a lot of tools which can do it based on sequence alone, but that's not our tool, right? And it's a supervised machine learning model. Show you how this works in a moment. So when we do the training, we take all available, all is again considering sequence identity and so on. So we build a subset of protein ligand complexes from PDB. Then imagine that you have a surface of the structure and you throw like a fishnet over it. The fishnet has edges, and on these edges, these are the points we consider. So these points, it has the fishnet has a regular spacing, and on each point on this regular spacing, we check the parameters or properties. So it's not connected necessarily with an amino acid as such, but it's a point in space. And we can, based on the structures, label these points as binding or not binding based on whether they are close to the ligand or not, right? And then we can extract vectors of physical-chemical parameters and structural features for each point and build a model. And it's a random forest model. We have about 30 attributes. The details probably not enough time to go through them, but that's what we've seen. And we have also added recently a conservation part of the pipeline where we check whether these residues are conserved in evolution, right? So for each, and again, you can use it in the pipeline, so that's why I'm showing this. And the idea is that if the residue is conserved, it's more likely to be a relevant binding site residue. Okay. And then when you do so you build a model, train it, and then you can use this model for any other structure, experimentally solved or predicted, right? And again, you can throw the net over the structure, find the points, collect their properties, and you can, based on these properties, you can cluster them as binding or not binding. And then if there are many points which have like binding affiliation, then you can cluster them into a binding pocket. So that's in a nutshell what this program does. And you will end up with something like this. You have a surface of the protein, and most of the points are green, meaning these are not binding residues. But here in this region, you get a number of red ones, which the algorithm predicts as the binding residues. And based on these points, you can build a pocket that is then shown to the user. So that's in a nutshell our program. But we always, you know that you can predict anything with limited amount of data, but always question whether it works, right? And again, you can show on many different metrics whether something works. And this can be like residue-based metrics, and that's important for this task. You can have either residue-based metrics, so you define residues which are supposed to be binding, again, based on the distance, for example, from the ligand, you define a set of residues which are binding residues. Or you can have a pocket-based metrics where you define the pocket and then you have very different set of parameters. And some methods are trained to come up with the residue-based, with the residues. Some are trained to come up with pockets, which makes it really difficult to compare them in the end, right? And so what P2Rank was trained on was to find the pockets. So it's trying to find the pockets. So the residue level characteristics are a bit difficult to apply to it. But what we try here is that you have top n plus k pockets, which should be the right ones for this. Why? So you could say, I'm interested in top 3 pockets. The K means that if the protein is binding more than 1 ligand, which is quite common, so it's binding 3 ligands, then we would not look on first 3, but 3 plus 3, so on 6 pockets, right? And then you test it, and then you have criteria like distance from the center of the pocket and so on. Again, just give you a... What has happened here? Okay, and then you have data sets, and that's the interesting part. You have data sets where you can test whether this thing works. And there are traditionally the ligand binding site prediction is quite well-occupied field, and there are a number of data sets which are used. And some of them, like this Chen and Coach, are used quite often by many tools. So then you can say, oh, I use it on as well, and then I can directly compare with the other methods, which is extremely useful. But you see that these test sets are quite small, 200 proteins, here are 400 proteins, 4,000 structures, okay, but not big enough. And that's where our tool was tested as well. And if you do that, you get to nice numbers, like in 72% of cases, you find the best pockets, which is better than other programs. Similar number on the larger data set, although already before. And this looks good, and we always thought it's probably too good. But then new data set was developed, and it was published actually last year by Javier Borges and Jeff Barton from Dundee, called Legacies, which is much more comprehensive. It covers about 65,000 structures with ligands. And if you look at these legacies, then from 60% we get roughly to 20% with P2Rank. It's still on top of the game, so there are not much better tools than this, but you see that from something which looks like the problem is solved, we see that we have a long way to go. The problem here is that Legacies is having a lot of interactions between ions and proteins, and P2Rank was not trained to do this. We know it cannot do it because it was not trained to do it. And it's large proportion, maybe 20%, maybe even 30% of the data set. But that's again how difficult these comparisons are. But I just wanted to show that we are aware that there's a long way to go. And so that's P2Rank. I will show you in a moment how we use it. I think that the main advantage of this program still is it's really easy to use. And you always, I said you start with a structure, but nowadays we often have many structures to start with. So I just want to give another brief intro into another tool which we have developed and you might think about when you will be doing this sort of experiments. And that's our tool to detect all the structures having the same binding site. And why we do that? Because we started this project because we thought the results from P2Rank are just way too good, and we just don't believe it is so good. And we thought one of the reasons could be that we are always testing on structures that are already binding a ligand. The ligand is taken out, but all the details of the geometry of the side chains is there. So we said, okay, let's go and start testing on the apo structures which are not binding the ligand. And we thought, I always told this to Christos, my student, that we don't do things because they are easy, but because we thought they would be easy. And this was one of the cases we thought, okay, so let's get the data set of apo structures and run it, and we will see how it goes. In the evening, we will have the results. That was not possible. We couldn't find the data set of apo structures. And just, this is going to work. Yeah. So just to get an idea what I mean with apo and holo structure, you have a structure of a protein binding heme, and the red one is binding heme. The blue one is exactly the same protein without a heme bound, right? And that describes the binding site, and you see that for the red one, the binding site is well formed, all the side chains are around it. But for the blue one, when you will try to like superimpose the ligand in there, you will see there's a lot of steric clashes. I think it will come in a moment. Here you see the pocket for the ligand in the holo, and here you see there is no pocket in the apo structure, right? So sometimes the details of the binding for the ligand can be very different depending on which structure you take. So that's again, what structure shall we take when we do these experiments? If we can choose, here is another example. These are the previous one, also the case of this sort of cryptic binding site. Cryptic binding site is a site which appears in the holo structure, but in the apo, it's not there. So you see here a hydrophobic molecule, which is a long pocket in the holo structure, but if you look at the apo structure, there is nothing there. All right. So sometimes we want to know what is apo and what is holo structure, whether they differ. And to do that, we have this tool and the database called AhoyDB, where you can search either by a protein structure or UniProt ID or a ligand name, and you get all the structures which are binding. And that's an important distinction I would like to stress. You can define apo and holo for a protein, but it's not accurate, because if you have more ligands binding the same protein, then the apo-holo is just not describing the complexity. It would be something like apo-holo, holo-holo, apo, or something in the most extreme cases. So we try to define this apo-holo not for protein, but per binding site. And that way you can get a list of structures in a given binding site, which are apo, which are holo, what is the ligand binding there, and some characteristics of how much they differ. We could go into this, but we will not have probably time, maybe later when we will be trying these things. And so to conclude all this is to give you questions rather than answers. One is, how do we test the quality of pocket prediction task? I tried to show you that you can do it like a pocket-based or residue-based, and there are so many characteristics. So it's always, I would say, you need to know what you want to predict, decide. This might navigate you what is the best criterion for you. And then what structures shall we use to do that? Nowadays, you can have AlphaFold structures. Do we know whether the AlphaFold structures are more apo or holo, or what the hell is the conformation of the structure? Most of the time, we don't. So it might be good reason to, for example, do homology modeling, the old days homology modeling, because there you can choose template and you know what will be the conformation of the binding site. And then I will show you, I have shown you the AHOIDB. And with that, I give this to Anna. She will tell you a little bit about MOL, and then we will show you how to use the programs. Oh, yeah.
Afternoon, my name is Anna Špačková, and I would like to introduce our tool MOL Online shortly. In modern drug design, we often focus on active site that can be on the surface of protein structure, but more often they are buried inside of protein structure. And the active site is joined together with outer space by tunnels or pores. And these tunnels can affect which molecules can go through the tunnels and then consequently react with this buried active site. First case of channels could be tunnels going from the buried active site to the outer space, and typically they could be, for example, for P450 family, enzyme family, and they are useful for transport some substrate in enzyme or so. The other case is pore. Pores usually occur in membrane proteins. They transport, for example, water or some ions. And to our tool, which is more online. It is a web-based tool for analyzing tunnels and pores in protein structures. Users can use PDB or MMC file for load the protein structure, and then MOL using Voronoi diagram for computing surface of the protein structure, and then cavities are analyzed. And MOL try to find out endpoints of the channels. The web page look like this. As I said, users can use an MC file or PDB file, or upload the protein structure directly from PDBE. And what is good to keep in mind, the users can select assembly, and if he or she can use a symmetric unit. After submitting the protein structure together with tunnels, if they are available in ChannelsDB database, are loaded into MolStar viewer. And then users can compute their own tunnels or pores using their properties. And tunnels are also available in 2D map. This map shows the tunnel structure. The tunnel is divided into layers, by lining residues. And in every layer, properties are computed depending on the lining residues. And that's all from my side. And now I will
go back. And now we will do the experiment with our server. We realized today there have never been so many users at one time. So let's see what will happen. We will do a short demo. You can do it with us. 5. We have very many. So what I would like to start with is this. So let's say we have a protein we want to look at, and I have chosen KRAS, but I will briefly show you how this works, and then you can try whatever you want, whatever you are interested in, and we are here to help you if anything is needed. So let's start with KRAS. That's like one of the main targets of drug design for very many years, because that's a protein which is mutated in roughly like 20% of all cancers. And it's a small protein which was for a long time thought that it's really difficult to drug it. And so we look at the KRAS, and that's one of the proteins. If we look at structures in UniProt, you will see that's one of those that has quite many structures. Actually, probably hundreds of structures available. So which one do we use, right? So let's first start with choosing ApoHolo, finding what this is. I copy the UniProt code, go to apoholo.cz, like this. I hope you can see that. And you get to this, and then to AhoyDB. Just type the code here, the UniProt code. And you can limit the search for X-ray structures or NMRs, exclude NMR structures, or define the threshold of resolution if you want to be more specific. But you can skip all this as well and let it run for a while. And you will see that we have something like 302 PDBs, and there are 12 unique uniprot IDs that might be a bit confusing if we chose just one, but it's because the protein is often in a complex with other protein. So it detects also the other proteins in the complex. And we know it's a small GTPase. KRAS is a small GTPase, so it should bind GDP and GTP, right? So let's find a place where it binds GDP. For example, GNP. GNP is an analog of GTP, which is very often used. I want it like this one, GDP. It has decent resolution of 2, so we can have a look here for APR. And we see that we've got quite some APO structures and number of hollow structures. And now, so that's our source of information. And now we can go to P2Rank. I will come back to this in a moment. P2Rank. If you type PrankWeb.cz, you will get to the place. And I said it's a simple to use program. I quite stand by this. Yeah, okay. That was not ideal. Like this, PrankWeb. And what you need to do is you need to know the PDB code of the structure. So if I would go back here, I said for APR, sorry, for EPR. And then you can decide to use the conservation pipeline or not use conservation pipeline. It's a bit faster without it, but our experience is that the conservation pipeline improves the results in terms of sorting the pockets better and providing often less results which are more relevant. So I would keep it. If you would want to have another structure, you can have a custom structure, so you can upload any structure you want, including predicted structures, or you can go directly to AlphaFold. You can use the AlphaFold from AlphaFold database code if that's easier for you. But we can start with this one and then just type submit, and that's it. So there is not a long learning curve doing this. We have just one pocket in this case, so that makes it easier. If you choose different structure from the list, you will see that it often finds more. And now here's the thing with the docking. So here you see the predicted structure. There is a score. If we have more, you will see that the score is decreasing. It's not easy to interpret the score or connect it with something real. We still struggle ourselves what the score means, but we know that it can quite well separate the hits we have. And you can zoom into the pocket by this, and you can, crucially with the new version, do a certain task on the pocket. So I can go create, so here you see the list of residues which are identified as part of the mining pocket. And you can go from here back to Ahoy. So these two tools are integrated, and search with this direct pocket in Ahoy if you want to. But that's not the task we want to show at the moment. So we have basically 3 different tasks. One is volume. It will just quickly calculate the volume of the pocket, so you can compare different pockets. let's say from closely related species, whether they differ in the size of the pocket. And then you can do mole to search for tunnels, and you can do the docking. I will show you now the docking. So if we go for docking, it uses Autodock Vina. So it's like very, very fast docking. And the thing is that you need to know what you want to dock in there. So it's up to you to say, this pocket is going to bind GTP, right? Because we would expect that if it's GTPase, it will bind GTP. So this tool is not telling you what it's going to bind, but you can do an experiment here, and that's when I can
But here it is possible just one structure to dock at a time.
Yes, it's just one at a time. And so let's do the GTP, for example. You need smiles to define the ligand. We know that this might not always be unique, but that's what we use at the moment. Are we here? Yes. So here you type, I would say GTP. And then here is a parameter which Autodock Vina uses, exhaustiveness, to describe how hard it will try to find the pose, how much computational power you give to it. So we go, the standard is 8. They recommend to use 32. We recommend to use 32. So we can go with 32. You do create task, you see it's queued, it's running. And we will see if we try it, many of us, we will see how quickly we get stuck or whether we can break it. We would be actually quite interested to know that. And I happen to have the results for different structure with the same task, which I tried here. It was different structure, but still of the same protein. And I got few pockets predicted. I can show you here. So it's actually quite sensitive, and you would see that there are few pockets predicted. And in this case, the protein is dimer. I will try to show this. It's a little bit, you can see a little bit. So the first 2 pockets are like identical pockets on different chains of the same protein. And what I tried here in the task was that I tried to dock GDP and GTP to the pocket, to the original native pocket, and then I also tried to dock it in a wrong pocket to see whether I can see the difference. So if I do that, here is docking into a wrong pocket of with GDP, and you see the energy -6. If I go here with the GDP in the right pocket, do I have? Oh, here it is. There's the GDP in the right pocket, and you see the energy is much better. So we believe it can help you to discriminate. You might know that this protein is binding this ligand, but you don't know which pocket. With the tool, you can identify the pocket and just try it. It seems to be able to discriminate. And if you bind GTP, which is here, GTP, if I'm not mistaken, should bind stronger than GDP, right? And it really seems so. It has still better score here, even though I'm docking into a structure which is binding GDP. Small GTPase, when it binds the ligand, it makes a movement in 2 loops, which improve the binding of both GDP or GTP. So you can go back to Ahoy.
Just a question regarding that docking, because you use those phosphate residues, and they dock in which state? Protonated, deprotonated? They affect the output.
It binds in the state which is in the structure.
Okay.
So it's very rough. It's just, I wouldn't say this is a way to dock a small molecule into a structure. It's more a way to make explorations about the binding sites that were identified, right? But still, so one question is which structure to use. And here, if you would use, and I think I have tried it, and you might try it as well, if you would choose one of the structures which is binding GNP, that's the one which should be more fine-tuned for binding GTP, and you would dock into that one, you should get a better score for GTP than into the structure that is binding GDP. So you can try this sort of task as well. And you can also try to dock into the APO structure. I happen to choose one where I even couldn't find any binding site on the APO structure for the GDP. I believe there are others where you can find it, but it shows how sensitive these structures are to these things. So it really matters which structure you choose, and that's probably more important than which program then you would use to find the binding sites, right? So these are this type of problems you might check: binding into a wrong pocket versus a right pocket, binding two different ligands into the same pocket, binding the same ligand into different structures of the same protein. And all this I believe, can be quite interesting. And then we have another task, which will use the tunnels. If I go back here, we can create task tunnel, and not on this structure because it doesn't have tunnels. But if we would use this one, I will. Yeah, okay. I will show you the results. So we have a protein which is cyclophilin, which is binding heme inside. You can see the heme here. And the heme is quite deeply buried. That's actually one of the big problems for P2 rank. If you throw a net on the surface, you might easily miss the binding sites, which are small and inside of the protein, because the net will simply not see them, right? But in this case, it can find it, even though it's very deep. But then you have question, can I get there? Can I get the molecule there? And in that case, you can use mole here and check. And in this case, you see there is pretty good tunnel going straight into the binding site. And again, you have some basic parameters like radius, average radius, length. This gives you some idea like what sort of molecule can possibly get there, although they are quite flexible. So these are the types of tasks you can try to do. So I just suggest that you try it with the structures you are interested in. And let's go and break the server. See whether we can make it. That would be quite useful for us to know which load we can take. Okay, do you have any questions to this? There are so many things. So you can download, of course, this is Molstar, the viewer we are using. And you've probably heard some rant about Molstar. That's probably one of the least like consumer-friendly program there is in the whole world. And like changing anything in there, it's quite nightmare. So if you are not able to do that, and you would be one of like vast majority of the population of this planet, then you can use SpyMall or whatever you want. So whenever there is, you can download the results and see them in more friendly program, which we are used to, right? And the same for Ahoy, where we also use Molstar. We use it because it's very fast. So when you upload, like in the Molstar here you can have hundreds of structures laid on top of each other. And this is a small protein, but if you happen to do this with the virus structure or ribosome structure, you might get stuck. But there is always a way to download the results and look at them in something much more friendly. Okay, so that's what we wanted to show you, and then now it's up to you to try and play with it, and we are here to help. So you might, for example, try to dock GTP into a structure which is preformed to bind to GMP. It's actually quite interesting trick which crystallographers very often use. If you give an enzyme a substrate, you are not very likely to see it in the structure because it will be cleaved or used by the enzyme. So you have to use some non-hydrolyzable analog, which would be AMP in case of ATP, GNP in case of GTP. So if we would go through the results here, I believe there is no single GTP in the structure because it's so difficult to see it in the structure. But there is a lot of GMP. But you can try any other protein you are interested in and check whether it works for you.
Maybe someone has suggestion what to show for other participants. Which protein?
No?
Show if possible how to find, for example, allosteric sites on kinase.
Whether we can find allosteric sites on kinases? Well, that's a really challenging task. What we actually working on this at the moment, allosteric sites on kinases. So each person working with the structures has like one structure it's always using, which is yours. I always use this one too as RC. It's a Src kinase, accidentally, because it's a beautiful structure and it's one of the proteins I really worked with during my career is probably a strong word, but during my scientific attempts. And it's an interesting kinase because it has a kinase domain and then it has four regulatory domains. But in those traditional structures, you see two, SH3 and SH2 domain. And and then there is a unique domain which has never been seen because it's completely disordered. And there's a short SH4 domain which is binding to membrane, which is also not seen typically in the structures. And the structure can have this closed form where these two domains interact with the kinase domain, or these two domains can be flipped away, and then this kinase domain is more active. And here you see the binding site for ATP, and then you see here a few more binding sites, which have, however, much worse score, so they are less likely to be true. But I think this one actually is one of those allosteric sites. I'm not 100% sure at the moment. That's one of the things we try to do with Ahoy at the moment, is that if you have a list of structures which have the same binding site, then you can look which have a big difference among them in the other binding site. It's very indirect, it's very slow, but we think that we can use it to identify allosteric binding sites from available data. But at the moment, what you can do with P2Rank is that you have other sites which are identified and you can try them. Although in this particular case, I think it's more like this this CDKs, right, which have the allosteric site. Do you know the protein which has it? Because at the moment, I would say I wouldn't spend much time with other sites identified here.
I have no suggestions, just from the general knowledge suggested to something more complex and less complex.
Yeah, so from the sites you see here, it predicts 6 sites, and only 1 is actually known. I would think that maybe the green one, maybe the blue one, maybe even the yellow one can be actual binding sites, which would, in principle, probably be these allosteric binding sites because we know what the protein is doing. And we know that the kinases have allosteric regulation. But that's the thing, it works always much better if you have a very concrete question you want to answer, right? Because then you have a lot of external data to work with the structure and try to understand this. And a lot of these allosteric sites, and that's another caveat for our program, is actually for ions. And P2Rank at the moment, that's one of the things we would like to improve in the future, but at the moment is not trained to identify ion binding sites, and we know it's quite poor in identifying them. It sometimes finds them if they happen to have similar characteristics as some bigger ligand, like drug-like molecule, but very often they don't find them. So that's also one of the problems you might have in finding these allosteric sites.
But even ions which are integral part of a protein or cofactor.
I mean, like zinc or sodium or potassium, things like that. And we also know that many of them, and that's from the work we've done on this, many of these ion binding sites are deeply inside of the protein. Again, quite invisible for the program at the moment.
Does this binding of those potassium ions change the functionality of proteins?
Sometimes it is deeply important. Like in this, some of the ion binding sites, like this ATP, it would typically bind to zinc or magnesium ions. But this is already in the pocket. There is no other specific pocket. It's just maybe it will be even visible there.
I'm curious mainly about those you mentioned, potassium, sodium ions, how much they affect the functionality of proteins. Mainly what we observe is magnesium, zinc, iron, copper, other.
The molecules, but I think this magnesium, they would typically shield the negative charge of the phosphate groups, right? So it's almost, they are almost always there, even if you don't see them, as long as they were in the solution and the crystallization solution, they will be there. Sometimes they might. We've seen even like water molecules, not even ions, but even the presence of the water molecules can lead to order disorder transition in protein structure. So either you see part of the structure because there are 2 water molecules, and if they are not there, a specific site, you will not see a complete helix. It's just this part will be invisible in the structure. So really sometimes tiny details can lead to significant changes in how you see the structure.
Water molecules are mainly on the surface, or
some of them are inside. And that's also like sometimes they would participate on binding the ion. So we did an exercise with students on the structural bioinformatics course where they were supposed to look at the structures which bind paracetamol. And paracetamol in many proteins binds mostly through the water molecules. So when they tried docking with this, it wasn't very successful because the waters were not added into the experiment, right? So they often, so you would have 6 interactions with amino acids, but 5 of them would be through water molecules, which are placed nearby the ligand. So this might be the difficulty. And when we do the prediction in P2 rank, we take the water molecules out as well. So we take everything which is not assigned as atom, like proper atom, so belonging to amino acid sequence. Did it work? Have you tried anything? Are you waiting too long, or is it? It's fine. That's good.
So then how do I assess the authenticity or reliability of the prediction for the ligand? So if I have here the energy output calculated, like is there?
It will always be relative, I'm afraid. So you can compare, if you know the protein is binding ATP, but you don't know which pocket, you can try all the pockets. I think it should be able to discriminate the best one, the right one, based on the energy, right? If you have 3 ligands, it might tell you which of them is most likely bind into the pocket, but it doesn't say that the third one is also not binding, right? Unless it's like really crap. But even you saw even here when we docked into completely wrong pocket, where was this? Was here. It was in a very different side, and it still got not plus like positive energy. It was still negative. There's a drop from 10 to 7, but still, it's for other ligands you would never get such a value. So it's not for virtual screening, right? It's just to test some specific hypothesis, and often I find this like trivial, but people are often helped just by the fact that they can look at the complex, right? So it improves, it supports thinking about the problem if you can see it, even though it's not perhaps 100% perfect. We had the docking. Oh, here we had, maybe that's also interesting. I can show this as well. So we had docking into the of the heme in the cyclophilin here, and here that's thing which we cannot do. And in this pocket there seems to be another molecule apart from heme, this cyclopropyl benzoic acid, right? And at the moment we can dock just one molecule into the pocket, and you will see that when I give there the best pose as predicted by Autodock Vina, it actually is relatively different. The orange one is the predicted one from the original structure, and it obstructs, or it's in conflict with this molecule, but it doesn't see it, of course. It's not aware that this molecule is there. While if we would use some other, like even like the third one, it's much closer to the original structure. So that's another thing. Sometimes the best pose doesn't have to be the one with the lowest energy, but it should also, the other thing is that we shouldn't think about the crystal structure as like the only possible truth there is. Even these small molecules, they will have some flexibility within the pocket. They can probably have slightly different poses in which they can exist in the pocket. So we shouldn't get just too much carried away by how the X-ray structure looks like. But here you can see that perhaps there is a problem that if in one pocket or very close to each other, there are 2 ligands, and we are docking just one, it will influence the results. And also that perhaps not always the best pose based on the energy ranking is the right one. Yeah, and here's the thing that, especially for the membrane proteins, the membrane proteins are often drug design targets, right? You should think about the tunnels as well, whether the molecule can get in there, because maybe if you make a box like here, you might find a lot of molecules that will bind there, but many of them might not be able to get from the outside of the cell into this position, which is deep inside of the protein. So that might be like, again, useful. It's more, I think all of this is more like useful visualization tools than like real docking. Although, as you could see, it does interesting things. It does predict GTP binding better than GDP. It can discriminate binding in the right and wrong pocket. Okay, so that's it. Thank you very much. Yeah.
Can I save if I load my ligand into it, can I save it as PDB?
Yeah, sure. So if you download the docking configuration, download results, you can do that, and then you can visualize it in PyMol or something, and it will also show download the energies, right? So it will also download the text file. I would actually recommend it. This is a really quick and dirty way of doing it, and some people are perhaps like comfortable with MolStar. I'm not, so I try to avoid it if possible. Okay, thank you very much. It was a long day.